Hello everyone, I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to discuss about the cell wall of bacteria. So since we are understanding about the structure of bacteria, so one of the video lectures I have already loaded is you can see the structure external to the cell wall. So the second one is the major important part of the bacteria, that is the cell wall of the bacteria. So this lecture I have elaborately discussed about the even the microchemical nature of the cell wall of the bacteria. So the first one, before entering understanding the cell wall of the bacteria, let us understand a brief overview about the bacteria. So the bacteria is a unicellular microorganism. It is a single celled organism. So microscopic in nature, having a size of around 0.5 to 2 micrometer in nature, and we can view only with the help of a microscope. And moreover, it is a typical prokaryotic cell structure the bacteria exhibits, because the prokaryote itself, the term means that it is having a primitive membranous structure may be present inside the cytoplasm. That means it doesn't have a well organized nuclear membrane like nucleus. It doesn't be present in the bacteria. Generally the genetic material will be present in the cytoplasm itself, and moreover it doesn't have an very membrane bound internal organelles like mitochondria. Like that, it carries some vacuoles like that in the cytoplasmic site. So if you want to understand about the bacterial structure, that is the structure of the bacteria, we can study it in three layers. One is the bacterial cell wall, thus I have elaborately discussed in this lecture. Second, the structure which is present external to the cell wall, and third one is the structure which is present internal to the cell wall. So already I have shared the video lectures on the structure external to the cell wall as well as structure internal to the cell wall. You can see in my other videos also. So first we will see a brief structure of the bacteria. So the bacteria will have a cell wall, right? So the some structure will be present outside the cell wall, that is especially a flagella, right? Then a short hair like structure called fimbriae. Apart from the outer covering called capsule and the S layer will be present. And some structure internal to the cell wall. The major one is the plasma membrane and the cytoplasm will be there. Inside the cytoplasm we have a nucleus, genetic material, DNA, and small inclusion bodies and ribosomes will be present. So in my previous videos you can understand about the structure internal as well as external to the cell wall separately. So this one will dedicatedly totally about the bacterial cell wall. So if you see the cell wall of the bacteria, this cell wall will give a rigid structure to the bacteria. So this will present just outside the plasma membrane. So this rigid structure of the bacteria gives a particular shape to the bacteria, whether we can say this is a round shaped bacteria called as cocci or a rod shaped one called as bacilli. So this rigid cell wall structure only plays an important role about the shape of the bacteria. So the one major striking difference between the cell wall of the human as well as the cell wall of the bacteria is that the bacterial cell wall is generally made up of a particular chemical called as peptidoglycan. So that's why once we given an antibiotic, it precisely targets the bacterial cell without touching the human cell. So that's why the most of the antibiotics like penicillin, the site of action is generally the synthesis of this peptidoglycan, that is the synthesis of the cell wall of the bacteria. Thereby it kills the bacteria effectively without harming our human cells. That's why it is considered as a silver bullet against the treatment of the infections. So if we see the purpose why the cell wall is important for a bacteria, the function of the cell wall, generally it determines the shape. That's why I told you it is the foremost one, first one. The cell wall of the bacteria is determine the shape of the bacteria. So the shape, whether it's a cocci, bacilli or spirilla or any other shape, is determined by the rigidity of this cell wall of the bacteria. Second, the bacteria cell wall protects the bacterial cell from the osmotic pressure. So whether it's a gram-positive or gram-negative bacteria, the peptidoglycan generally prevents the bacteria from this osmotic shock, and it is generally more efficient with gram-positive bacteria. Once we understand the structure, you can understand how effective it is in gram-positive bacteria. And the third one is it prevents the entry of the toxic substance, even the antibiotics. So if you see some gram-positive bacteria like acid fast staining bacteria contains mucolic acid along with the cell wall, that is the high lipid content, will inhibit the entry of these antibiotics. Thereby it protects the bacteria from this toxic substance. And moreover, this cell wall helps in the pathogenicity of the bacteria. So for example, if you take a gram-negative bacteria, it carries a lipid A which acts as an endotoxin of the gram-negative bacteria. These are the major functions of the cell wall. So we can classify the bacteria into different types based on the cell wall. So this was first put forward in 1884 by Christian Gram. So he developed a technique called as Gram staining technique. Based on the response of this bacterial cell wall on its Gram staining technique, he has divided the bacteria into two major groups. One is the gram positive and another is the gram negative. So the gram positive bacteria are generally gives positive reaction to this gram staining techniques, and generally it takes up the primary stain. Most commonly used the primary staining agent, if you see, is the crystal violet. That's why the gram positive bacteria will look like purple in color. And the second one is the gram negative bacteria which fails the gram staining technique. So instead of taking the primary staining agent, it takes up the counter stain. That's why it looks like a pink in color. So what actually this gram staining technique is that we will treat the particular strain specimen with 4 reagents. One is the primary staining agent, second is the modern, third is the secondary staining agent, and fourth is the decolorizing agent. So the most commonly used the primary staining agent is the crystal violet, and the counter staining agent is the generally safranin, which is having a contrast color. That's why you can see a purple as well as pink color, both the positive and negative species respectively. So the first we take the specimen, heat fix it, then we will add the primary staining agent and grams iodine. After some times we will decolorize with 95% ethanol. Then we add the counter staining agent, then view under a microscope. If the bacteria takes the primary staining agent, then we call this a gram positive cell wall bacteria or gram positive bacteria. If it takes up the secondary agent and loses the primary staining agent, then it is a gram negative bacteria. This is how the bacteria has been classified based on this cell wall. So this I have shown you a picture. You can see that not only the coloring it is chemically and the structurally completely different, the gram positive and gram negative bacteria. So the gram positive bacteria, you have only 2 layers. One is the plasma membrane layer and second is a thick peptidoglycan layer. The space between this plasma membrane and the thick peptidoglycan we call as a periplasmic space. So if you see in gram negative bacteria, there is a plasma membrane. After that we can see a thin membrane of peptidoglycan, right? Then there is a space, then we have another a little bit thick membrane called outer membrane. That means the gram negative cell wall will have a 3 layers, that is the plasma membrane, peptidoglycan and outer membrane. The space between the plasma membrane and outer membrane we call as a periplasmic space, right? So this is the striking difference between the gram positive bacteria and gram negative bacteria. So the gram positive bacteria will be thick, gram negative bacteria will be thin. Gram positive bacteria will have 2 layers, gram negative bacteria will have 3 layers. Let us have a quick overview about this gram positive cell wall and gram negative cell wall. Then we can understand the chemical nature of both the cell walls. So the gram positive cell wall is majorly composed of a particular homogeneous layer of chemical called as peptidoglycan. Otherwise we can call as murin. It is thick in nature, around 20 to 80 nanometer thickness it has having. It just lies outside the plasma membrane. So because of this thick nature of this peptidoglycan, it is osmotically, gram positive cells are osmotically more resistant compared to gram negative cell. So the gram negative will have a thin peptidoglycan layer, the gram positive will have a thick peptidoglycan. That's why it is osmotically more resistant. So if you keep a gram positive cell under a hypertonic or a hypotonic solution, it doesn't get damaged. Otherwise, it will get broken away. So this is the major difference between the gram positive and gram negative cell wall. Then the second part, if you see that the space between the plasma membrane and the peptidoglycan we call as a periplasmic space. It carries many proteins. It is generally occupied with a substance called periplasm. This helps in the transport of the material. So this is a simple outline what we have seen. There is a plasma membrane, then a peptidoglycan layer, thick layer, between them a periplasmic space is present in gram positive cell wall. So in contrast, if you see a gram negative cell wall, a brief overview, the gram negative cell wall carries of 3 layers and it is quite complex in nature when compared to gram positive cell wall, because it carries away from the plasma membrane, it carries a thin layer of peptidoglycan layer around 2 to 7 nanometer thick and a little bit thicker layer of outer membrane around 7 to 8 nanometer thick. So the space between this plasma membrane and outer membrane we call as a periplasmic space, and it carries periplasm and some protein nature. So this you can see from the picture. So we have a plasma membrane. So there is a plasma membrane and the outer membrane, between that we can see a periplasmic space, right? So between this plasma membrane and outer membrane, a thin layer of peptidoglycan layer will be there. So the gram negative cell wall has 3 layers: plasma membrane, peptidoglycan and outer membrane. Between plasma membrane and outer membrane, we have a periplasmic space. This you can see from the structure of the cell wall staining also, right? So out of this, the major chemical is the peptidoglycan. So before getting into the chemical structure, I will see a brief overview of the difference between the gram positive and gram negative cell wall. Gram positive, we have a plasma membrane and a peptidoglycan layer, whereas the gram negative, we have a plasma membrane, a thin peptidoglycan and an outer membrane. This is the major difference that exists. Additionally, the gram negative will have an outer membrane which is absent in gram positive cell. So out of this, the major chemical we have to understand is the peptidoglycan, because it is present in both gram positive cell and gram negative cell. So in gram negative it is a thin layer, in gram positive it is the thick and the major layer. So the peptidoglycan, the name itself tells that peptido means it carries a protein and glycan means it carries a sugar moiety. So it is a polymer. So otherwise this peptidoglycan is also called as murin. It is a net-like structure. It is a mesh-like structure of polymers composed of identical units of glycan and the protein unit. So this peptidoglycan polymer contains majorly 2 sugar moieties and proteins also. So the 2 sugar moieties are N-acetylglucosamine, we called as NAG, and second is the N-acetylmuramic acid. It is nothing different from N-acetylglucosamine. It is simply a lactic acid ether of N-acetylglucosamine, N-acetylmuramic acid. So the 2 sugar derivatives are present in this polymer, NAG, that is N-acetylglucosamine, and NAM, that is N-acetylmuramic acid. Apart from this, it carries some different types of amino acids. So that's why it is called peptidoglycan. So glucose glycan part plus protein part. So the amino part of these polymers are generally 3 major amino acids will be present. One is D-glutamic acid, D-alanine and mesodiaminopimelic acid will be present. So it is generally dextrorotatory in nature because this doesn't interact with a peptidase enzyme. So to protect the polymer from the activity of the peptidase, the amino acids are generally dextrorotatory in nature because these peptidase can act only on the levorotatory isomers. So if you see from this picture, the first 2 sugar moiety, that is the NAG and the NAM. So N-acetylglucosamine, then N-acetylmuramic acid. Both are structurally similar in nature, only the D-lactic acid is added as an ether. So that's the one major striking difference between the N-acetylglucosamine and N-acetylmuramic acid. Then it contains L-alanine and the three major acid, D-glutamic acid, mesodiaminopimulic acid and D-alanine. So these three amino acids will be majorly present. Then additionally L-alanine is there. So the peptidoglycan is nothing but it is a polymer of NAG and NAM. In NAM, it is attached with 4 amino acids, that is L-alanine and D-alanine, then D-glutamic acid and mesodiaminopimulic acid. Right? So based on this peptidoglycan structure will act as a sac. So it works as a sac. So this can be isolated and we can study it effectively. So the presence of the mesodiaminopimulic acid, it differs from gram-positive and gram-negative bacteria. So generally the mesodiaminopimulic acid will be present in gram-negative bacteria. This has been replaced in gram-positive bacteria with L-lysine. So the presence of this L-lysine will produces a strong mesh-like polymer by forming an interbridges. So if you see some gram-positive bacteria, not only some, most of the gram-positive bacteria will have a peptidoglycan interbridge. That means a group of glycine amino acids around 5 to 10 will form a bridge between the carboxyl group of D-alanine and amino group of diaminopimulic acid or L-lysine. So this is the major one that it can form. This peptide interbridge makes the mesh very stronger when compared to other one. So gram-negative cell wall generally lacks this peptide interbridge. Gram-positive cell wall will have this peptide interbridge. This you can see from the picture. So if you see that, so in gram-positive bacteria generally the diaminopimulic acid can be replaced with L-lysines, or some bacteria contains the diaminopimulic acid. So the carboxyl group of the D-alanine and the amino group of the L-lysine, we get a peptide interbridge which is made up of glycine. So this will be seen in a gram-positive bacteria. That's why it is osmotically more resistant and the peptidoglycan layer also very strong in nature. Whereas the gram-negative bacteria, it doesn't find any peptide interbridge. It simply forms a bond between the D-alanine and diaminopimulic acid. That's all. So this is the striking difference between this peptide interbridge in gram-positive and gram-negative bacteria. So that's why the peptidoglycan will look like a sac. So it can be easily isolated away from the bacteria, and after isolation it maintains its integrity of its shape. It is but relatively porous and elastic in nature. So let us little bit understand the basic chemical nature of the both gram-positive cell and gram-negative cell in depth. So I have already told that the gram-positive cell is majorly made up of particular chemical, that is peptidoglycan. So the peptidoglycan concentration differs from gram-positive and gram-negative bacteria. So the gram-positive bacteria, we can see up to 50% of the peptidoglycan layer, and in gram-negative only 10% will be present. And moreover, the gram-positive bacteria will have peptide interbridge made up of glycine, which is not present in gram-negative bacteria. This you can see from the picture. So there is a N-acetylglucosamine and N-acetylmuramic acid is there. The peptide interbridge is present. So which is not present in gram-negative bacteria. So apart from this one, if you see that the peptidoglycan of the gram-negative bacteria will be very thin in nature, and I have also showed that the 50% of dry weight is made up of peptidoglycan in gram-positive, only 10% will be present in gram-negative. But in gram-positive, apart from this peptidoglycan, it carries one more chemical called as teichoic acid. So this teichoic acid is present in large amount in gram-positive bacteria, but it does not present in a gram-negative bacteria. So teichoic acid, chemically if you see it, it's a polymer of a glycerol or ribitol, which is joined by a phosphate group. Generally the amino acid which is present in the teichoic acid is a D-alanine, or sometimes a glucose molecule can be attached with this glycerol or ribitol. So it is a polymer of a D-alanine, glycerol or ribitol with glucose or without glucose, right? This teichoic acid can exist in two ways. One, the teichoic acid can be covalently attached to the peptidoglycan layer only. Otherwise, it can be attached with the lipid part of the plasma membrane. So if the teichoic acid is present in the peptidoglycan, then it is simply called as teichoic acid. If it is bind with the lipid part of the plasma membrane, then it is called as lipoteichoic acid. So the major function of this teichoic acid is it contains the magnesium ions on its surface, it binds with, and thereby it prevents the bacteria from thermal injury. So this teichoic acid, moreover, comes outside the surface of the peptidoglycan and provides a negative charge to the bacteria. So the gram-positive bacteria will shows a negative charge because of the presence of teichoic acid, and it is completely absent in the gram-negative bacteria. So if you see from this picture, you can see that the peptidoglycan layer is there. The teichoic acid which is docked in the peptidoglycan layer, otherwise it can be docked from the plasma membrane, then it comes out from the lipid part, it is called as lipoteichoic acid. So the second one, if you see the example of this bacteria carrying teichoic acid is the Staphylococcus aureus and Streptococcus faecalis. Apart from the teichoic acid, some bacteria carries other substances. The major one is the polysaccharides present in Streptococcus pyogenes, which can be isolated with dilute HCl. So this is the major chemical composition of a gram-positive bacteria. So if you see the chemical nature of a gram-negative bacterial cell wall, so before getting into the gram-negative bacterial cell wall, let us see one of the bacterial cell wall, that is the acid-fast bacterial cell wall. So the acid-fast bacteria are generally gram-positive in nature. So some of the acid-fast bacteria, if you see, it is a mycobacterium or corynebacterium, which is rich in lipids. If you see, generally the lipids are very less in gram-positive bacterial cell wall because it majorly contains peptidoglycan and teichoic acid and small amount of lipid. But the lipid is very high in acid-fast bacteria, and it generally carries a simple lipid structure, that is CHOH, CHR1 and R2, then COOH, which is an hydrocarbon in nature. So the major lipid part which is present in the Mycobacterium tuberculi or Mycobacteria species is the mycolic acid. So the mycolic acid derivatives are also called as cod factor, which is basically a chemical of trehalose dimycolate. It is toxic in nature and it is responsible for the toxicity disease to the humans caused by the Corynebacterium diphtheria or Mycobacterium tuberculosis. So the lipids will be high in nature in gram-positive cell wall. That's only answer to acid-fast staining. So one of the best example is the Mycobacterium species, which carries mycolic acid in the cell wall, right? So the second major one cell wall is the gram-negative cell wall. So if you see the gram-negative cell wall, I already told that it is more complex compared to gram-positive walls, even if we study chemically, because it carries a thin peptidoglycan layer after the plasma membrane, then a little bit thick outer membrane, then there is a periplasmic space between the outer membrane and the plasma membrane. So it only contributes 5 to 10% of the dry weight of the cell. So in this picture you can see we have a peptidoglycan layer, then we have a outer membrane, then we have a plasma membrane. So this we have a periplasmic space. So in this there are not many chemicals are there. Some called the bronze lipoprotein will be there. Some Porin proteins will be there. Some outer side chains are also present. So the most studied example of the cell wall of the bacteria is the E. coli. The E. coli carries only 2 nanometer thick of the peptidoglycan layer, which comprises of 1 or 2 sheets of peptidoglycan layer. So that's why the antibiotics are sometimes more resistant to gram-negative bacteria because it has a less peptidoglycan layer. The second major part which is present in the gram-negative bacteria is the periplasmic space. It's a huge periplasmic space is present, and this is the striking difference with the gram-positive bacteria. And this periplasmic space can range up to 1 to 71 nanometer, whereas there is no or little periplasmic space in the gram-positive bacteria. The periplasmic space are generally composed of periplasmic proteins. So the major role of this protein is the nutrition acquisition. It gathers nutrition from outside. For example, certain hydrolytic enzymes or transporter proteins will be present as a periplasmic proteins. This transports of the nutrient membrane. It hydrolyzes the polysaccharides and takes the glucose as a transporter protein. They are also involved in peptidoglycan synthesis. So it modification can lead to toxicity to the cell, also can harm the cell, right? And moreover, the periplasm carries hydrogen ions more. For the locomotion, the proton pump is needed for the rotation of the flagella. So the periplasmic space as work as an energy conservation part of the cell. For example, a denitrifying bacteria converts the nitrates to nitrogen thing. So this is the second part, which is next to the peptidoglycan. And the major part of the gram-negative bacteria is the outer membrane. So the outer membrane is the most complex part of this gram-negative bacteria. It is generally linked with the next to the thin membrane, and it carries the two things: bronze lipoprotein and Porin protein. These two things are present in the outer membrane, and the outer membrane is generally composed of a particular chemical called lipopolysaccharide. So you can see from this picture, the outer membrane is present, right? It is majorly composed of lipopolysaccharide, right? Then it carries a bronze lipoprotein and the Porin protein. So first we understand about bonds lipoprotein. So bonds lipoprotein is the most abundant material which is present in the outer membrane. So it is generally connected with the peptidoglycan layer. So it is covalently joined with the peptidoglycan layer, underlying peptide, then it goes through the outer membrane, and it is generally embedded in the outer membrane by a hydrophobic bond. So you can see this, the hydrophobic region, the bronze lipoprotein gets attached, but it is docked with the peptidoglycan layer, right? So the major chemical which is present in the outer membrane is the lipopolysaccharide. So the name itself tells that the lipo means lipid, polysaccharide means the glycan part. So gram positive cell has less lipid, whereas gram negative cell has more lipid because of this outer membrane, that is the lipopolysaccharide membrane. So it carries both lipids and carbohydrates. It is majorly composed of three parts. One is the lipid part, we call as a lipid A. Second is the core polysaccharide part, and third is the O side chain. So in this picture you can see there is a lipid part, right? So we call as a lipid A. Then a core polysaccharide part which is majorly composed of 10 sugar units. Then a O side chain part which acts as an endotoxin also. Sorry, lipid A acts as an endotoxin. This is acts as an antigenic part also. So if you see the lipid A part of this outer membrane, that is the lipopolysaccharide, it contains 2 glucosamine sugar units that I have shown in the pictures, and it carries 3 fatty acids connected with a phosphate group. So this is the lipid A, which is generally projected outside the outer membrane. So this lipid A is often toxic to the humans. Generally this is works as an endotoxin. So if the bacteria destroys, lyses, the cell wall gets depressed. Out of this, the lipid A acts as an endotoxin and produces the symptoms of infection of the gram-negative bacteria. The second is the core polysaccharide, which is joins the O antigen with the lipid A, which is generally made up of 10 sugar units. And the final one is the O side chain. It is also called as O antigen. It extends from the core to the outside. So in the picture you can see it is freely flowing outside, and it is responsible for the immune response, for stimulating the acquired immunity. The O antigen of this bacteria is very important. So you can see the lipopolysaccharide, the O side chain is slightly projecting the outside, which works as an antigenic part. So this is the important part of the gram-negative bacteria, and the outer membrane is very responsible for producing this negative charge, especially the lipopolysaccharide, and it helps in the attachment of bacteria to the host tissue and causes pathogenicity of this bacteria, and by overcoming the host defense mechanism. And moreover, it acts as a permeability barrier which helps in the transport of food materials from outside to inside. It also restricts some toxic chemicals to the bacteria like the antibiotics, toxic substances which can harm the bacteria. And moreover, the second protein which is present in the lipopolysaccharide layer is the porin protein. So it is a permeability barrier which generally helps in the transport of small molecules like water or glucose like molecules can be transported easily from the outer membrane to the inside the cytoplasm. So this porin protein, if you see the structure, it is basically made up of a trimer. So trimer means it is a 3 protein structure carrying a hole, so like a tube like structure which allows the transport of low molecular weight compounds which is having a molecular weight of around 600 to 700 daltons. So if we see this porin protein here, so it is a trimer carrying holes for the transport of these proteins. So I think this lecture will be very helpful for you of understanding the cell wall of the bacteria. So thank you very much. So in my future lectures, you can understand the various aspects of the other structures of the bacteria, especially the structure external to the cell wall and the structure internal to the cell wall. So this lecture has completely given an insight about the cell wall of both gram-positive bacteria and gram-negative bacteria through its morphological characteristics as well as the chemical aspects also. So thank you very much.